My name is Jane Carlton. I'm a professor at New York University. I first started on malaria when I was an undergrad, actually. So I was at the University of Edinburgh, and they had a large malaria group there. And that malaria group was one of the first places where you could actually do genetics of malaria parasites. So you can take a male malaria parasite and a female malaria parasite and actually do genetic crosses and then look to see how the genes associate between the different what are called progeny. And I thought this was fascinating, and I was really thrilled about this and decided to join one of those labs as my undergraduate project. And then basically after that I was just bitten by the bug, I guess, and wanted to stay on to do my PhD, which I did in the genetics of malaria parasites, and then it just took off from there. Primarily because I'm a geneticist, and these days geneticists actually are genomicists. So instead of looking at individual genes, you now look at whole genomes. And when I finished my PhD, the whole area of genomics was really up and coming, and I thought this was an area that could be used to interrogate the biology of malaria parasites. So I was one of the team that decoded the whole genome of Plasmodium falciparum, which is the most common malaria parasite found in Africa. And then more recently, I decoded the genome of Plasmodium vivax, which is the most common species outside of Africa. And that's also taken me into this whole area of the monkey malarias. Monkey malarias are actually a fascinating clade. Monkey malarias are found in Southeast Asia, and they can infect humans, in particular Plasmodium knowlesi, but they are also bona fide parasites of monkeys in their own right. And because they share this evolutionary history with Plasmodium vivax, we can actually learn a lot from the monkey malaria parasites that will enable us to inform us about Plasmodium vivax and how we might find better ways to cure the malaria caused by that species. I really take my hat off to Drs. Mary Galinsky and her colleagues for bringing together all these different disparate types of biology and disciplines which are really required for what's called systems biology. And what I bring to the table is my genomics background. So having sequenced a range of different Plasmodium species, I really have the knowledge to be able to decode, annotate, and analyze a lot of genome sequences. So right now we are sequencing the genomes of Plasmodium coatnei, which is a great model system for Plasmodium falciparum, and also Plasmodium inui and a couple of other monkey malaria parasites too. Actually, another component of one thing that we're doing is I lead a large 7-year Centre of Excellence for Malaria in India, and we will be providing samples from this particular Centre of Excellence, which can then be fed into all of these different projects which are part of MaHPIC. And so I'm very excited about that because it's basically leveraging what we're already producing in one centre for the good of MaHPIC. And so we can hopefully get double the amount of information and really contribute to this fantastic systems biology approach to the study of malaria. As part of these isomers, I have to spend at least 30% of my effort out in India. And so myself and several members of my team actually flip-flop across the Atlantic. We're very good friends of United Airlines, and we spend a lot of time in India, which I love. It's a fascinating country, full of smells and noises and bustle, but fabulous people, wonderful food, and it's a great pleasure to be hosted in that country, especially working on a particular disease of such global health importance. Malaria eradication, the E word, is an interesting one. I think in particular, perhaps some people have thought that the main focus for eradication should be on falciparum malaria because that's the malaria that kills the majority of people. However, from my studies and other studies, we are starting to understand that actually vivax is going to be the more difficult parasite species to eradicate. And so you'll basically be only solving half the problem if you just concentrate on Plasmodium falciparum. Plasmodium vivax eradication is just as important, I feel. So MaHPIC, as it does, bringing together all these different disciplines, including looking at host-parasite interactions, I think is going to produce a whole wealth of data for us to be able to try and eradicate the genus as a whole, not just concentrating on falciparum, but also vivax and potentially Malaria and Ovale as well.